Good evening, everyone. Thank you very much for coming. We are so pleased that the weather cooperated this evening, somewhat, at least I should say. It's slowly warming up, and we're pleased that you are able to join us tonight. Welcome to the David C. Hardesty Jr. Festival of Ideas at West Virginia University. For more than a decade, the Festival of Ideas has enriched students' education and broadened their thinking on the day's most important issues. As a WVU student leader in the 1960s, David Hardesty organized the first Festival of Ideas. Nearly 30 years later, he returned to his alma mater as its university president, and in 1997 relaunched the festival as an annual event, drawing renowned speakers to campus every year. Festival speakers have come to our campus from around the world, from diverse backgrounds, and from right in our own backyard, featuring some of our nation's most brilliant thinkers. Following tonight's lecture, we will take questions from the audience, and one of our student ambassadors will come to you with a microphone. We encourage you to share your thoughts via Twitter using the hashtag WVUideas. Tonight we feature one of WVU's own star pupils, Paul Holcomb, PhD candidate at the WVU School of Medicine and researcher in the Center for Neuroscience under the direction of Dr. George Spiro. Paul participated last spring in a very friendly competition among graduate students and faculty called Science Idol, sponsored by Science on Tap and the WVU Department of Biology. In it, contestants were challenged to present a 5-minute talk on a complex science topic in an engaging way to a broad audience. Paul's talk, Making Connections, the Science of Wiring the Brain, was judged to be the most effective and the winner. I should note, Paul's wife, who is here with us tonight, Assistant Professor Mickey Holcomb, came in second place in that competition. The results may have been different if it had been a different evening, but obviously they're quite a dynamic duo, and we're excited that they're both here with us tonight. Paul is back to demonstrate his skills and research with a full-length presentation entitled The Brain Initiative, Computers, Connectomes, and the Emergence of Technobiology. As a researcher in the field of connectomics, Paul's talk will outline how researchers aim to tackle this large-scale problem, the technology that is needed, and what scientists hope to gain, especially in the areas of learning and memory, stroke recovery, and treatment of neurodegenerative diseases such as Alzheimer's and Parkinson's. Paul Holcomb is originally from Charleston and attended South Charleston High School. A graduate of Vanderbilt University, his research interests include brain-machine interfacing, development of the auditory system, neuroplasticity, and computational neuroscience. He is currently working to obtain a PhD in neuroscience, as well as a certificate in STEM education, both here at WVU. Please welcome tonight Paul Holcomb.
Thanks, everybody. It's wonderful to see so many people here tonight. I would like to point out that the first thing that my wife told me when I got back to the table after winning Science Idol was that I shouldn't beat my wife. So, which I agreed with wholeheartedly. I would like to thank the Festival of Ideas folks for putting this on, as well as the Science Idol folks and the organizers of Science on Tap for giving me this opportunity. Now, the first thing I'd like to do tonight is actually change the title of my talk. Now, don't worry, I'm not changing it much, just a little bit. I would like to replace technobiology, which we'll talk about, with neurotechnology. And the reason that I want to do this is I think that one of the major things that you can get out of this talk is the fact that neurotechnology isn't something that you just see in science fiction movies. It's actually something that's affecting individuals right now, and that's going to have a tremendous impact on our society in the future. So whether you know it or not, right now we are living in the century of the brain. What do I mean by that? In the 21st century, we are going to learn more about how the human brain works than in the rest of human history combined. And that sort of innovation, that sort of ideas, are going to fuel the technology that's going to affect our lives in the next 10, 15, even 20 years. So what is neurotechnology? Well, you can think of neurotechnology in 3 different ways. The first way is devices that allow us to peer into the functioning human brain and get ideas on how the brain works. With those ideas, we can then create devices that are implanted or interact with the human brain, which can create happier, healthier human beings. I had to work our kids in here somehow. They're definitely happy, healthy human beings. Another way of looking at neurotechnology is the intact brain functioning with a device, not implanted, that can make normal everyday tasks easier for all of us, whether we have a disease, a disorder, or not. So let's take an example of a neurotechnology that's actually been used recently, and that is deep brain stimulation. Deep brain stimulation is a recent innovation that actually involves implanting an electrode into the brain, which is connected to what's called a pulse generator, which creates electrical signals to stimulate the brain for a desired effect. You can think of this almost as a pacemaker for the brain. And deep brain stimulation is being used to treat a host of diseases. Anything from Parkinson's disease, which is the major target for deep brain stimulation right now, to chronic pain that doesn't respond to normal treatments, normal drugs, severe depression, and even Tourette's syndrome. Now, it's one thing for me to stand here and tell you that deep brain stimulation and other neurotechnologies are affecting lives. It's another thing entirely to actually see it for yourselves. So I'd like to show you a video of a gentleman that has Parkinson's disease. And one of the key symptoms of Parkinson's disease are uncontrollable tremors, the inability to create very precise motor movements. This gentleman has had a deep brain stimulator implanted, but we're first going to see him with the deep brain stimulator turned off. And as you can see, he has severe tremors, inability to create those fine motor movements like we were talking about. Now they're going to turn the deep brain stimulator on through an external device. And what we'll see is how profoundly his Parkinson's disease is impacted by this deep brain stimulator. After the deep brain stimulator was turned on, the gentleman could touch his nose, could move his hands, could even move individual fingers with absolutely no tremor whatsoever. And this was moments after the deep brain stimulator was turned on. So this sort of technology is, as I said, not science fiction. It's actually impacting people's health today. So how does deep brain stimulation work? How does neurotechnology work? Well, in order to understand that, I think we need to first start with some fundamentals of the brain. How does the brain work? This is a human brain. It's roughly 3 pounds. All of us have one. And based on mainly damage to specific areas of the brain in past patients, we've learned a little bit about what each area of the brain does. For example, there's an area that's designed for vision, for hearing, for creation of motor movements, for sensation, taking in thermal sensations or touch, for understanding speech and forming speech. So later on tonight, when you ask me questions, hopefully I'll be able to use this area to understand your question and also hopefully be able to formulate some answer that makes sense. There's also another section of the brain involved in decision making. This is sort of the higher order thinking that we believe is unique to human beings. And the decision making area is particularly important in things such as telling me not to make incredibly complex PowerPoint slides. So we've looked at sort of the macroscopic version of the brain, but what is the brain made of? Well, the brain is made of cells, just like any other organ in your body, but they're made up of very specific cells. There are 2 main classes of cells in the brain: neurons, which are the signaling cells in the brain that create circuits that allow you to do the myriad of functions that you do. And another class of cells called glia. Now, glia literally means glue. When we first discovered these cells, we believed that they were nothing but brain glue. They held all of the neurons together. Over decades of research, we've actually found that these glia form a myriad of functions. Unfortunately, although glia are one of my favorite cells in the brain, I'm not going to actually have time to talk about them tonight. Find me afterwards and we can talk at length about glia, I assure you. So neurons and glia are split roughly 50-50 in the brain. But how many of these cells are there? Well, recent estimates show that there are roughly 200 billion cells in the human brain that are split roughly evenly between neurons and glia. Now, put that into perspective as the sheer size of that number. There are roughly 300 billion stars in the Milky Way galaxy. So you're approaching that with the number of neurons in your brain. And not only that, but in terms of complexity, each of these neurons can create up to 10,000 connections, which calculates to about 100 trillion connections in the human brain. It's a lot to keep track of. So right here, I would like to address my first brain myth of tonight. I think that one of my responsibilities as a neuroscientist is dispelling myths about the brain that have been held for a very long time by the general public. For instance, humans are smarter than other animals because our brains are bigger. Now, who here before tonight believed that this was true? Can I get a show of hands? I won't judge you, I promise. Okay. And it's actually a reasonable assumption, because if you have more brain, you can do more stuff, right? Well, let's take a look at our brain in comparison to some other brains. So we have the human brain here, obviously larger than the cat and the dog and the gorilla brain. Well, now look at the dolphin and the elephant brain. If this were true, dolphins and elephants should be as smart, if not smarter, than humans. And in fact, the smartest animal on Earth should be the blue whale. Now, no offense to the blue whale, but I think we've probably got them beat in the intellect department. So what defines intelligence? Well, the short answer is we really don't know. But there is a better metric than just the size of the brain to determine what might or might not be intelligent. It seems that a ratio of the body to the brain mass may actually be a better indicator of what is and isn't intelligent. For instance, most mammals have about a 140-to-1 body to brain mass ratio. You can think of this as 140 pounds of animal to every 1 pound of brain. For humans, we have about a 50-to-1 body to brain mass ratio. So it may be that it's not the sheer size of the brain, but the size of the brain compared to the size of the organism that makes a difference in terms of intelligence. So getting back to our neurons, neurons aren't just one type of cell. They come in many different shapes, many different sizes, many different functions. And different areas of the brain have different types of neurons, and they connect with other types of neurons. So it's not just a single cell, but this complex relationship between multiple types of neurons that form the human brain. There are, however, some similarities between these cells. So if this is a simplified neuron, all neurons have a cell body, which you can think of as sort of the factory of the cell that produces all of the things that the cell needs to stay alive. And then processes that are specialized for the function of the cell. Dendrites and an axon. And these dendrites are designed to receive signals. The axon is designed to transmit signals. So you can imagine that if you had a chain of these cells, each one could receive a signal and transmit it to a neighbor, who then transmits it to their next neighbor, and so on and so forth in a circuit of cells. What does this look like? that we had a bunch of these neurons, and these neurons were all plated into a single plate and allowed to grow and connect with one another. What we would see is individual neurons that light up when they fire and connect to other neurons who also light up in sort of a chain reaction of events. And studying this chain reaction, studying how these neurons connect with one another, is the field of connectomics. So connectomics basically encompasses the study not only of the individual cells in the brain, but also how they connect with one another. Now, why is this important? Well, as we saw before, we know a reasonable amount about what these individual cells do, how they function. And we believe we know a lot about larger structures in the brain and how those structures function as well. The problem is we know very little about what happens in between. We don't know nearly enough about how we can take these cells and turn them into this brain. Or as Sebastian Sung would point out, Sebastian Sung is a prominent researcher in connectomics at MIT. How do your billions of dumb neurons turn into a smart person? So the goal for connectomics is really to create a wiring diagram for the brain. Similar to a wiring diagram for a house, where you can track back a particular wire and find what it connects to and how it causes that thing to work. We want to be able to create a wiring diagram for the human brain. Now, obviously, this is not the human brain. This is actually a very special worm called C. elegans. And it was actually the first connectome ever to be completed and published. And why is this worm so special? Well, it has 302 neurons and about 5,000 synapses. And each of these neurons in each C. elegans is exactly the same. Every time a C. elegans is created, it's created with the same 302 neurons in the same locations with the same connections, which means that once you map out one C. elegans connectome, you know what all C. elegans connectomes look like. Made it a very powerful tool. Unfortunately, human brains aren't like that. My connectome does not look the same as your connectome, does not look the same as her connectome, which means that we have to map out multiple connectomes, multiple wiring diagrams in order to start to get at what makes us human, what makes us work. It's a very daunting task. Now, my second brain myth that I'd like to talk about now actually feeds directly into that. One of the myths that I've heard quite often is that we only use 10% of our brains. It's used quite often in science fiction, where they want to show that we could have telepathy or other mental powers by being able to tap into the 90% of our brain that we don't use. Well, this is my brain. And I had it scanned mainly to prove to my wife that it's there. And this is what it would look like if we only used 10% of our brain. This would actually be great for connectomics, because we could throw out 90% of the brain, 90% of those 100 billion neurons, and only focus on the ones that are really important. Unfortunately, this is not the case. So this is refuted by brain damage. So you can imagine, as we saw before, that there are different areas in the brain that are involved in different types of sensation, different types of function. If we only used 10% of our brain, you could damage 90% of the brain and still be a completely functioning human being. And we know that's not true. This has also been refuted by brain imaging. We now have the capability, using functional MRI, of looking at the brain as we're performing tasks and actually quantifying how much of the brain we're using for any given task. Now, I would like to show you a video here, but I probably won't be able to. We will test it out. This is a mouse brain using a new technique called light sheet microscopy. And all of the individual lights that you see here are actually firing neurons or groups of neurons in the brain. Now, did you see that spike just then? The mouse was actually presented with a stimulus which caused the vast majority of the brain to activate. I'm glad that worked. I really liked that video. And what I want to point out here is that if a mouse is using more than 10% of its brain, I put to you that more likely than not, humans are using more than 10% of our brains. So the truth of the matter is we most likely use most of our brain all of the time. And this is actually kind of unfortunate for connectomics researchers in that we're brought back to the fact that we have to map 100 billion neurons and all of their connections. So how do we map connections in the brain? Well, there's a gold standard for doing this type of research currently that we actually employ here at WVU. Now, we don't do it on human brains. We do it on mouse brain. But I'm going to step you through at least some of the steps for doing this. We dissect out a small portion of the brain, and we use a technique called serial block face scanning electron microscopy. Now, I won't go into too much of the detail of the technique, but what I will tell you is that with this serial block face scanning electron microscopy, we're able to create incredibly large data sets that still allow us to track very small processes over long distances. So we do this, we gather many, many images, thousands of images, and we stack these images back together to create, basically in virtual reality, something that approximates the brain that we've just sectioned. Now, I would like to show you a video of how this works. So what we see here is not in time, but in depth. We're moving through the brain of the mouse. You can see a blood vessel, which is a large piece that just went off the screen, and small cells that continue to come in and out of the screen. Now I'm going to stop it here. What do we do with this data once we have it? Well, currently, we have teams of tracers that go in with tablets and pens and actually trace out the cells themselves on every single slice of the tissue that we have. And you can see that there's a problem here. If we want to get a large number of cells, we need a large number of tracers or a lot of time. I think it's important to appreciate just how detailed this data is. We're zooming in very close to look at individual parts of the cell. There's a significant bottleneck with the way that we accomplish connectomics right now. This technique is the gold standard, but unfortunately in some ways it's sort of the lead standard because it is so incredibly slow. I did a calculation for you to just demonstrate how slow this could be in terms of the goal of creating the connectome of the human brain. So it takes me roughly 60 hours to reconstruct a single neuron in the mouse brain. If you extrapolate that out to the human brain, it would take me roughly 1.37 billion years to reconstruct the human brain. It's hard enough to get a grant for 2 years, let alone 1.37 billion years. So obviously we need different methods for doing this. And that's where technobiology comes in. And what is technobiology? It's the application of technology to biological problems. And it's not a new concept. This has been occurring for just about every great biological revolution in the history of biology, beginning with the microscope allowing us to see that all living things are formed from the same cells. The ability to amplify DNA so that we can sequence it, or the personal computer, which not only allowed us to create the models that we saw or would have seen if the video had worked, but also to create complex models in virtual reality, and almost more importantly, communicate between scientists over vast distances so that we can share ideas. So what kind of technobiology do we need as connectomics researchers? Well, one thing we would like is an automated tracing system. That is, taking an image, handing it off to a computer and saying, find all of the neurons in this image. Getting something back that looks somewhat like this, where the computer has found all of the edges for all of the things in that image, and then having it define what is and isn't a neuron within this image. Now, this software is just in its infancy, and unfortunately it's not as good as we would like it yet. We take for granted the fact that we can look at this image and find individual objects within the image, because our brains are optimized as pattern recognition generators. We can find edges in this image much faster than a computer can, and with much higher accuracy. And the more cells that you give the computer to find, the worse it is at finding them. So this sort of technique with automated tracing actually requires a lot of user intervention at the back end, which in the end takes almost as much time as just tracing the things yourself. So not as effective as we would like it yet. Another technique, which was developed by Sebastian Sung at MIT, who I mentioned earlier, is EyeWire. And they thought, you know, a single person tracing a single cell is going to take a long time. Well, how about we create something that people are going to want to play that will allow thousands of people to trace cells? Hoping we're not also having a fire alarm now. All right. So what is EyeWire? EyeWire is actually an online game that allows people to sign up, go in, and begin tracing cells, defining what is and isn't a part of a cell. They've recently surpassed 100,000 users for EyeWire. Now, I wanted to tell you tonight how many cells Sebastian Sung and his group for EyeWire have managed to reconstruct. So I got on the website to try to find this, and I never actually found that number. But I spent about an hour and a half playing the game, and it was a really fun game. So if anyone is interested in getting into science in a meaningful way, I would suggest going on and finding EyeWire. It's completely free to play, and you're contributing to very important research. So the US government, about a year ago, realized that things like EyeWire, things like automated tracing, things like connectomics, neurotechnology is the wave of the future. And so in order to foster that, they created the BRAIN Initiative. And as Obama says, the BRAIN Initiative is designed to unlock the mystery of the 3 pounds of matter in between our ears. So what is the BRAIN Initiative? Well, the BRAIN Initiative stands for Brain Research Through Advancing Innovative Neurotechnologies. There's that word again. And it's slated to cost about $100 million a year over the course of 10 years. Now that might seem like a lot of money, but if you consider that there are about 314 million people in the United States, according to the census, that That calculates to about 32 cents a person, which is less than the cost of a postage stamp. And it has the potential for an immense return on our investment, both in healthcare as well as economically. So to examine this, let's take a look at one of the projects that the BRAIN Initiative is very frequently being compared to, and that is the Human Genome Project. The Human Genome Project, which started in 1990, ended in 2003, is a 13-year project, whereas the BRAIN Initiative is currently slated for about 10 years. It costs the United States $3.8 billion, about 4 times what's slated for the BRAIN Initiative right now. The return on investment, as calculated, $796 billion. Another way to look at that, for every $1 we put into the Human Genome Project, we got $141 back out. Not only that, but the Human Genome Project is projected to have created over 310,000 jobs that we could sorely use right now. So in terms of comparing the Human Genome Project to the BRAIN Initiative, if we can invest and get the type of return that the Human Genome Project gave us, we will have a much stronger economy and also a much stronger healthcare industry. So who's involved in the BRAIN Initiative? Well, there are 3 different organizations that are collaborating for the BRAIN Initiative. The National Science Foundation, DARPA, which is the Defense Advanced Research Projects Agency, and the National Institutes of Health. And each of these organizations wants to get out something slightly different from the BRAIN Initiative. The National Science Foundation wants those neurotechnologies, those cool new techniques and devices that we talked about, to apply not only to neuroscience, but to basic sciences. DARPA, who's tasked with keeping the United States military on the forefront of technology, wants technology that will allow soldiers to be more effective, protect them more, and this technology will hopefully trickle down and affect the general population. The National Institutes of Health, who is charged with the health and well-being of Americans, want not only cures for neurological diseases, but also to understand fundamentally how these diseases come about. What sort of diseases are we talking about? Well, we talked about Parkinson's disease earlier in regards to deep brain stimulation. Alzheimer's is a second disease that they're interested in. How many people have had a friend or a family member that have been affected by Alzheimer's? A great majority of you. It's a terrible, debilitating disorder. And it impacts not only those that are affected, but also their friends and their family members in a very serious way. And unfortunately, we have a lot of theories about why Alzheimer's disease occurs, but no real concrete idea of why it occurs. And Alzheimer's disease rates are actually going up. So combating this disorder is paramount to neuroscience research right now. Concussion. Concussion is a hot button topic in sports right now, especially hockey and football. And we know that damage occurs during concussion, but we don't really understand how long-term that damage is and what occurs when there are multiple concussions. And we're not talking just about physical damage to the brain. We're also talking about mental, emotional, and even developmental disorders that we know very little about. Schizophrenia, autism, ADHD, Tourette's syndrome, a whole host of different disorders that we could potentially begin to understand if we can unlock how the brain works normally. That's a key point. If we can understand the functioning of the normal human brain, then we can begin to unravel what happens when things go wrong. Right now, though, we need to focus on determining how the normal human brain works in order to be able to get at these diseases. and to improve the quality of life for these patients. And we can also think about the impact of improving the quality of life of these patients in terms of an economic standpoint. So let's take Alzheimer's, for example. Last year, $203 billion were spent in the United States on Alzheimer's care. $203 billion. Imagine if we could invest $100 million a year, or even $1 billion over the course of 10 years, and save $203 billion, as well as tens of thousands, if not hundreds of thousands of lives. I think that's a very worthwhile investment. Talking about Alzheimer's brings me to my third brain myth, and that is, as we age, our brains don't work as well. We seem to have this idea that as we get older, we're supposed to start forgetting things. We're supposed to not be able to take care of ourselves at some point, not be able to hold conversations or perform our day-to-day activities, and that is not true. The World Health Organization has stated that dementia, and I would add in Alzheimer's disease, Parkinson's disease, and other disorders that are associated with aging right now, they are not a normal part of aging. In fact, you can have people that have lived 110 years that can still hold perfectly normal, incredibly intelligent conversations. And conversely, we have people that are 30, 40 years old that are being diagnosed with early onset dementia. So dementia and these other disorders are not necessarily caused by aging, but by some other factor that we don't understand yet. There was also a study in 1992 that looked at the brains of centenarians, that is, people that have lived over 100 years. What they found was that those brains looked very similar to people's that were 30, 40, even 50 years younger. So our brains are not getting older, they're optimizing. So getting back to our neurotechnology, I'd like to leave you tonight with a sense that, as I said, neurotechnology is not science fiction. It's actually something that's happening right now that is going to impact all of our lives in the next 10 or 15 years. Hopefully my videos work, we'll see. The first technology I'd like to talk about comes from a company called Emotiv Systems in San Francisco. And Emotiv Systems has developed a technology that is capable of completely non-invasively reading out the function of the brain and transforming that function into a myriad of different uses. The Emotiv Systems device is called the Insight. And what it's capable of doing is, without any gels, without any invasive procedures, it can actually read electrical signals from the brain. You can input them into a computer, and you can do anything from driving a car without using your hands to composing music simply with your own thoughts. It has tremendous implications for rehabilitation. Emotiv is now in their second generation device called the Emotiv Insight. And they've started a Kickstarter for this. Kickstarter, in case you don't know, is actually a website where people can get on with ideas and attempt to get funding. And they wanted to raise $100,000 for establishing this device. And they raised $1.6 million. So what this tells me is that even in its infancy, neurotechnology is exciting people. And more so that there are people out there that see the need for neurotechnology and are willing to fund it. The second neurotechnology I'd like to talk about is from a company called Second Sight, the Argus 2. And this has actually been in the news recently due to the fact that the FDA just approved this as the first retinal implant. It's designed to replace damaged retinal cells, which are the light sensitive cells within the eye. It utilizes a camera, which is mounted on these specialized glasses, that send wirelessly a signal to the eye, which then transmits that signal back to the brain to be processed, just as though you're seeing through the camera. And this has tremendous implications. They've actually been able to restore sight to those that have been blinded by genetic eye conditions. In a similar vein, the idea of neuroprosthetics has been around for a while. I actually, as an aside, this is what really sparked my interest in neuroscience and in my previous incarnation as a biomedical engineer. And I remember seeing Luke Skywalker in, I guess it was the 6th Star Wars movie now, although it was the 3rd when I saw it, lose his hand and come back with this robotic hand. And I thought, that's amazing. That would be great for us to do. Why can't we do that? Well, as it turns out now, we actually can. I'm going to attempt this video. I can't guarantee that it'll actually work. Excellent. So what you see here is a gentleman who's lost his arm and has been fitted with a replacement arm from a DARPA project aimed at being able to replace limbs for limbs that have been lost in the line of combat. Now, what's important to point out here, and besides the fact that the arm functions almost identical to a normal arm, is that it is completely controlled by the user's brain. Previous arms such as this have used different actuators, have used pedals in a foot that control the arm. In this particular case, all this gentleman needs to do is think about what he wants to do with the arm, and it occurs. And similarly with the DARPA projects, there's a new project that they've recently introduced called the DARPA RAM project. RAM stands for Restoring Active Memory. The goal of this project is implanting a device into soldiers such that if they're injured on the battlefield and they lose abilities such as driving a car, tying their shoelaces, being able to brush their teeth on their own, daily things that we take for granted. This device, having stored those types of memories, can then return those memories to that person. Now this is, as I said, in its infancy in terms of the research necessary to do this, but this is one of the projects that DARPA is actively pursuing right now. And you can imagine that this has implications beyond the battlefield. For instance, a more advanced version of this device could be implanted into Alzheimer's patients and allow them to store memories, which would then be accessible to them as their memories deteriorated. So I'd like to close tonight with what I think is possibly the most important initiative goal, brain initiative goal. And that is sharing knowledge and training. The Brain Initiative is not set up solely to train neuroscientists, not solely set up to train scientists in general, but is also designed to inform the public about what it is we're doing as neuroscientists and how it can benefit you. And that's actually part of why we're here tonight, the Festival of Ideas. There are also talks online called TED Talks, which if none of you have gone and seen TED Talks, I'd seriously recommend them. They're short talks given by prominent scientists on a variety of topics that are designed specifically for a lay audience. Also, next week is Brain Awareness Week. Brain Awareness Week, which occurs every year, is designed to have neuroscientists move into the community, discuss with middle schoolers, with high schoolers, with community groups, what we do, how their brains work, discuss brain health, brain wellness. And I think all of these things are incredibly important, not just in the view of the BRAIN Initiative, but in the view of science in general. Communication is key. If we want to move forward as a society, as a race, as the human race, we need to communicate more about what we are doing. And just to take the neuroscience perspective, neuroscience can't move forward with neuroscientists alone. We need doctors to tell us the symptoms that they're seeing in patients with neurological disorders, to tell us the progressions of these diseases. We need physicists to uncover the fundamental aspects of the technologies that we need in order to do neuroscience. We need engineers to design devices that are usable by neuroscientists. We need mathematicians, computer engineers, software engineers to be able to design software that will dramatically increase our ability to do things such as connectomics. We need politicians to go to bat for us, to allow us to have the funding necessary to take on incredibly complex problems, like what is Alzheimer's disease? How do we prevent autism? And we need you. We need you to go out into the community, to share your stories, to tell people about friends or family members that you've had that have suffered from neurological disorders. We need you to go out and tell people why you think spending your 32 cents a year on the BRAIN Initiative is an important thing to do. We need you. Thank you. Any questions?
At this time, we'd love to take your questions. We've got two ambassadors here with microphones. If you'd like to just raise your hand. Do you have a question for Paul?
Sure.
We've got one right here. She'll come to you with the microphone.
Yeah, we can hear.
If you were able to do what you want with neurobiological technology, would you be able to, I guess, reverse the process of aging? Like an 80-year-old person could go play basketball if they wanted to, as if they were in their 20s?
So aging is a far more complex question than just neuroscience. There are so many facets to aging that go well beyond neuroscience. Do I think that it's possible to reverse aging? I think that we don't know enough about aging yet to say whether it's possible to reverse aging. Do I hope that we can reverse aging? Sure. I'd like to stay in my 20s forever, but...
I guess what my question is neurobiological technology only a means to help people think more clearly as they age and stuff like that?
No, so there are many potential applications for neurotechnology. One that comes to mind right away is in paraplegia and quadriplegia. We may be able to repair the damage that's done in the spinal cord to paraplegics and quadriplegics, such that they can either communicate with their limbs again, or designing things such as exoskeletons, which I've recently seen a completely 3D printed exoskeleton that was used to allow a famous skier that had been paralyzed from the waist down to walk again, simply by using the exoskeleton along with electrodes that were implanted above her waist. So there are neurotechnologies that are capable of doing quite a bit of things. You need to remember that your nervous system isn't just confined to your head. Your nervous system is pervasive throughout your entire body. It's how you interact with the world and how the world interacts with you. And so any way that you can think of that you might be able to alter that, there might be a neurotechnology for that.
Other questions?
is there potential for dealing with addiction with implanted devices? And the second question, if we can control, and I understand, set the implanted devices for Parkinsonism by remote units, what is the potential for controlling people who have implanted devices?
So I'll address the first question first, and that is that there's a lot of research being done in addiction right now, and we believe that addiction is actually involved in multiple areas of the brain. Once again, I unfortunately have to fall back to the fact that we don't really know enough about the brain, and especially the addicted brain, in order to be able to treat as of yet. But, as I said, one of the goals of the Brain Initiative is to create the neurotechnologies necessary to be able to see what's going on in the brain in the cases of addiction. Do I believe that eventually we'll be able to basically invasively treat addiction? It's possible. We're treating things like chronic depression right now with deep brain stimulation, so it's possible in the future we might be able to treat addiction as well. Whether or not we'll be able to treat it effectively is another question. And in terms of deep brain stimulation and being able to control an individual with deep brain stimulation, you have to remember that these electrodes, at least right now, are incredibly small, and they're placed in very precise locations. So, for instance, in order to control an individual, you would have to place multiple electrodes throughout their entire motor cortex to make them walk sort of like puppets. And even then, you wouldn't be controlling what they think. You would only be controlling what they do. So it's unlikely that through deep brain stimulation, you would be able to control an individual. I'm not, however, going to discount the idea that there won't be neurotechnology developed that could control an individual.
Other questions? We'll go to this side here.
Yeah, Paul, I have a question about idiot savant or savant, where people, after accidents, for example, they develop some ability to do something unusual, right? You must have heard about it. So, you know, 10% versus 90%. Is it possible we can tap into some dormant, you know, potential of the human brain? I wouldn't say send people to crash, but, you know, if we find
Right.
which part of brain is involved, maybe we can make this, you know, we can make out of people who are normal.
Enhance that part of the brain.
Right, enhance, exactly.
It is possible that there are areas of the brain that do particular things well. It's also possible that people that have been injured in such a way that they can't do things that they normally were able to do, instead take up things that they would have been good at otherwise, but didn't take up because they were too busy doing other things. For instance, if I were paralyzed and I had always wanted to play the piano and I had extra time on my hands, I might take up the piano and find out that I was in fact a savant at the piano. Is it possible from a neurological perspective that because I had been damaged, I then became a savant at the piano? Yeah, sure. Our brains remap in situations of damage quite frequently. And it depends upon a host of factors: age, the status of the brain in terms of the amount of damage of the brain, rehabilitation, as to whether or not the brain can remap. And I would think that in that case, the brain might be remapping in sort of an artificial or incorrect way that would unlock this sort of potential in the brain. But I would argue that that potential was probably already there to begin with and was just not being realized by that person.
Other questions? Here?
The body tends to reject foreign objects that are placed into it. I was wondering what the success rate is for these technologies being placed in the body, and also if the body does reject the technology, how bad could that be? Could that cause the technology to malfunction?
So I would worry far more about what it would do to the body than what it would do to the technology. We've been getting much, much better at creating substances, creating actual materials that are more what we call biocompatible. So they have less rejection by the body. You also have to realize that the brain itself is actually an immunoprivileged area. So it is sealed off from the rest of the immune system in the body for the most part. And so it is possible that we can implant things in the brain that, if we were to implant them somewhere else, might cause severe rejection. But in the brain, because you have this sort of closed system, it may be able to survive, and you may be able to have no long-lasting effects. Now, there have been problems with degeneration when you put implants, for example, into the brain. But in terms of deep brain stimulation, since it's a fairly new technology, there aren't studies that go out for many, many years in order to see whether or not that's the case. So I guess we just have to wait and see.
You mentioned remapping, but you didn't talk about what might be myth number 4, that brain cells can't regenerate. At least that's what I remember learning when I was a kid. But I think I've heard recently that that is not actually true, that maybe we can regenerate parts of our brain. So I want to ask you about that, but I also wanted to say, please keep DARPA out of my brain.
You give me too much credit with my influence with DARPA, but I'll try. In terms of being able to regenerate brain cells, we do know that brain cells can regenerate to a certain extent. There are specific zones within the brain that contain cells that can proliferate and turn into neurons. But those cells are relatively rare. They're very difficult to find. I've seen experiments trying to find them, and it takes several animals, many animals, to be able to find this niche of regenerative cells. And in terms of large-scale damage, one of the problems that you actually run into has to do with my favorite type of cells, which are the glial cells. It has nothing to do with the ability of the neurons to regenerate, but rather the job of these glial cells, one of the jobs is to move in and encapsulate damage such that it can't spread to other regions. And once it does that, it forms, similar to a scar that you would get on your skin, it forms a glial scar in the brain. And that scar is maintained throughout the rest of the life of the individual. So in terms of targeting regions of brain damage or spinal cord damage, you have to target very early to get regeneration, because if you miss the window where that glial scarring has then taken place, it's very difficult to get beyond that.
Hi, Paul.
Hello.
My question has to do with the technique that you employed in your research.
Mm-hmm.
And the technique that I'm thinking about is scanning microscopy.
Mm-hmm.
My understanding of scanning microscopy is that you use it when you want to examine the surface of a cell or surface of a tissue.
Mm-hmm.
And you avoid sectioning.
Right.
And what it does is it eliminates the artifacts which you introduce. Because of sectioning, you have to harden the tissue in order to section it.
Right.
So if you were looking at the sections in order to superimpose them to build the structure of the tissue, why didn't you do transmission microscopy?
So that actually gets into the details of serial block face scanning electron microscopy. So you're right. A traditional scanning electron microscopy, you either section a tissue and scan that tissue, or you scan just the surface of a block of tissue. One of the major problems in previous experiments trying to do what's called serial section electron microscopy, where you take multiple sections and you scan each individual section and try to stack them back up, is one, user error. If you have a slice of tissue that is hundreds of times thinner than the human hair, and you attempt to handle it, it can tear, it can fold, you could drop it, and at that point you've lost one of the images that you need in order to be able to reconstruct whatever it is you're trying to reconstruct. Another problem is if you're scanning tissue on a slice-by-slice basis, all of those slices aren't going to be in the same orientation every time that you scan. A slice can rotate, a slice can move over slightly, and because of that, it's very difficult to get the slices aligned properly when you try to stack them back up. With serial block face scanning electron microscopy, what we've done is actually integrated what's called a microtome inside the electron microscope. And what that allows us to do is take a block of tissue, put it inside the microscope, never handle it after that, scan the surface of the tissue, cut off the surface of the tissue and throw away that slice, and then scan the surface of the tissue again. The tissue hasn't moved, so every image that we get is aligned with the previous image. And we don't have to handle any slices. All of the slices that we're cutting have already been scanned. We're just throwing them away at that point. So that allows us very quickly to build these sort of three-dimensional blocks of tissue with very high fidelity in terms of alignment between the slices.
Okay, we'll do our final last three here. One in the back there. Go ahead.
I had a question about the bioprosthetics. So I'd heard early on in people with debilitating strokes, in order to control a cursor, they would have to think of discoordinated thoughts. You know, so with the arm, you'd have to think, if you wanted to go lower, you'd have to think of a sunset, or, you know, you couldn't actually control it with the thought of wanting to move your arm. Is that device cortically mapped to like a motor?
Yeah, so it's actually mapped to not necessarily the motor cortex, but it utilizes the nerve signals to muscles that are left within the stump that the device is attached to. So you can imagine that there's still neuromuscular associations within that area. And those signals are still intact. You could still tell your arm to do something, even though your arm isn't there. And so what this device does is reads out those signals. It's actually through implanted electrodes in the stump. It reads out those signals and translates them into what it believes your motion is being computed as. It believes you're trying to do, and then translates that into the actual motion. So there's no actual cortical mapping necessary. There is some training in getting the signals correct, both from a computational standpoint and also from a user standpoint, but it's nothing like the discordant thoughts that are necessary for certain types of cortical integration, like with the stroke patients.
Let's take them on this side first.
Hi, I enjoyed the talk. I wanted to go back to the question about the microscopy method. And among the major players here, you had NSF, NIH, but is the National Institute of Standards and Technology being engaged in this effort for development of reference materials and reference models? And then going a step further, you know, one of the keys to the success of the Genome Project was standardization of the data collection, the modeling, the reporting. Are there efforts to manage the informatics? Because you're going to generate a lot of data through this initiative.
Yeah, most definitely. There are efforts, not necessarily through the National Institute of Standard and Technology, but internal efforts through places like the NIH for making the data both standardized and also available to all researchers. One of the key aspects of the BRAIN Initiative is transparency, is our ability to share the data that we collect with a group of researchers that may be able to extract something from it that certain other researchers might not be able to. For instance, if we're only looking at neurons and someone else wants to look at vasculature, and we have this data already, there's no sense in having them go back and recollect the same data just to look at something that we already have. So there is definitely a push for standardization and for centralization of information. You have to remember that in terms of size, these data sets are incredibly large. We're dealing with data sets that are on the order of 2 to 3 terabytes of information right now for actually relatively small areas of the brain. So as this gets larger, as this balloons, then we're going to have a very important data collection issue to address. But I think that as we progress in the BRAIN Initiative, we're also going to have progressions in other types of technology that are going to allow us to both store and transmit data much faster.
Two more. Last 2 questions.
I could stand up here all night.
How far are we from telepathy?
Ooh. Now you're really getting into science fiction. So I would say fairly far. Telepathy is not necessarily just a neuroscience problem. You're talking about fundamental physics, in which case you'd want to talk to my wife and not me. Do I think that the human brain is capable of interfacing either with objects or with other brains? Yes. Do I think that we're at the point where we can do that now? No. We don't even understand enough about the human brain to know why it breaks, let alone what else we can do with it. But that would definitely fall under that sort of third aspect of neurotechnology that I talked about, where the brain interfaces with something outside of the body in order to make things easier for us. I'd love to be able to sit on my couch and grab a drink out of the refrigerator without having to get up. But I don't think it's going to happen anytime soon.
All right, we'll take our final question here in a second.
Thank you very much for your talk. The two research areas that have been widely researched in the past 10, 15 years for assistive technology are the electric stimulation of the muscles through the brain signals. And the second one is the wearable robotic area, which has been, in the literature in the past 10 years. Well, the third one that you just unveiled was a deep stimulation of the brain. How do you see the future of these 3 technologies, and how do you think they can help each other, and how do you think the neurobiology or neurotechnology can help them to make a better life for people?
That's a very complex question. I'll try to answer it as simply and as quickly as possible. So similar to what I talked about with the paraplegia and quadriplegia, I believe that a correlation between technology external to the body, such as exoskeletons, the ability to accurately read out electrical signals both from the muscles and from the brain, and then the ability to interface with the brain directly in order to repair damage, are all going to sort of coalesce into this almost integrated system where you can take a person that, say, is a quadriplegic, outfit them with a suit, and allow them to regain all of their motor function. And in fact, I don't see that as being very far off. I think that the more we learn, especially about motor function, the more likely it is within the next probably 10 years we're going to see something like that. Ideally, in a case like that, you don't want to be invasive. You don't want to do something like what we saw in the deep brain stimulation. If you can stay as external to the body as possible, then you'll have the maximum user base. Because let's be honest, people probably are going to be a bit resistant to having things implanted in their brains. And looking back at the pacemaker, when the pacemaker was originally created, people were very hesitant to get pacemakers implanted because they were being implanted in their hearts. Now, pacemakers are relatively commonplace. So I think with more education, with more communication about how these devices work and what the potential benefits are to these devices, then we have a greater likelihood of being able to help more people by having a culture of acceptance of neurotechnology. Yeah,